Welcome to the third part of best practices for hit generation. So here we're going to look at what we call hit validation. And one important thing to note is that even if we placed it within the classical screening strategies, what we're going to talk about here is actually valid for pretty much all hit generation strategies. So in the previous webinar, we looked at screening approaches which led to the identification of hits. Here, we're going to develop how we validate these hits to make sure that they really modulate the target of interest and with an attractive mode of action. And this is extremely important because this will enable us to make sure that we select the right compounds for chemistry optimization. So in order to validate the hits, we have to put in place what we call a screening cascade. And that is a suite of assays, additional assays and filtering steps that will enable us to actually select the right hits, the validated hits. Now, the reason for this is that in the original screen, we used a simplified biological system that was needed to screen large amounts of compounds, especially for high throughput screens. And it's important that we confirm in a physiologically more relevant setting, that is probably lower throughput, that the compounds still work as we think they should work. The other important point here is that compounds with apparent activity in an assay do not necessarily engage the target of interest. So they may be active, but they may show activity through another mechanism than actually engaging the target you're interested in. And these could be of two kinds. One is false positives that have an apparent activity in the assay, but that's an artifact. Or they could be compounds with undesired mode of actions. And of course, we want to remove those. So in a way, we want to have a cascade that is looking like something in the picture here, where we have multiple checkpoints. And at each step, the hits are tested. And we discard the ones in red that are either false positives, have undesired properties or MOAs, or that don't work in biological or disease-relevant assays. Each step will then only retain the interesting compounds, and at the end, we will have our validated hits. Now, this slide here shows a prototypical screening cascade. And of course, this is just an example. You really have to think about designing this cascade carefully based on a particular target. So we saw in the previous webinar that we run our primary assay on 10,000 to millions of compounds. We confirm the hits. The next step was IC50 determination. So we ran a concentration response curve in the same primary assay. Then we usually go into filtering and triaging the compounds. So it's important to have a compound purity check because the impurities could actually be responsible for the activity you see. And on the right side, what you see is we will go into clustering, meaning putting together compounds that are similar, but also triaging them based on properties like log-D, and flagging them if they are frequent hitters or contain pain motifs. We're going to go into that in a little bit more details in a minute. After we've done this, we're going to run them through additional assays. And there are two types of assays which we will develop a little bit in more details. The first type is counterscreens. These are assays where you don't want your compound to be active in. So compounds active there will be discarded. And orthogonal assays, which are assays where you want your compounds to be active, and only the ones that are active will be carried forward. And finally, we're going to run them through so-called secondary assays, which are more complex assays with high disease relevance. And again, we want the compounds to be active there. At the end of your screening cascade, if you've designed it properly, the hits that you have should be validated. Now, there's a couple of things to note. The first thing is the order of steps can depend on the throughput of the assays and the number of remaining hits. So ideally, you would start with assays with the highest filtering efficiency, because they will remove as many unwanted compounds as possible. Now, it's sometimes difficult to know a priori which order you should use. So it can be useful to actually test the flowchart, the screening cascades, with hits from a small diversity set and reference compounds if you have them. And remember, in the previous webinar, we advised to run a pilot screen. So you can use actually the hits from this pilot screen to do that. The other point to note is shown on the right. Depending on your hit rate of the primary assay, you can have different numbers of compounds. So usually you hope to have below 1% hit rate, but the hit rate can be much higher, above 10%. And if you talk about a million compounds, you can imagine that you will not be able to run all of the hits into the more demanding assays downstream. So depending on this, you may want to be more selective in your clustering and your triaging. The last very important point is that at the end of the flowchart, at the end when you think you have your validated hits, it's very important to resynthesize and purify these hits for final confirmation. There are numerous examples of small impurities which you don't see that were causing artifacts in the assays, or even of compounds, samples which had the wrong structure assigned to it. So let's go into a little bit more details now. We talked about clustering. Clustering means put together similar compounds. The idea then is that you can reduce the effort either already at the stage of the dose response curve or for the hit validation. If you have a very large hit list, you may not be able to test all of them, but you can select representatives of each clusters, and that will be sufficient to actually get an idea on the whole cluster. You can also already extract early SAR and prioritize these clusters based on the properties or potency distribution. Finally, it can be helpful to identify singletons early. So singletons are compounds which don't have near neighbors. And if the structure is of interest, maybe you want to test analogs that may not have been tested already. There are a variety of methods with which you can do clustering. We won't go into details here, but there's a very interesting review cited at the bottom of this slide. So we talked about how important it is to remove false positives as early as possible from your hit list. So to understand what a false positive is, it's important to think about that the effect of a compound in an assay is not always linked to the desired target engagement. It can have many other origins, and some of this is explained here. First, it can come from the compound structure and the sample quality. Impurities, as mentioned, like heavy metals or trifluoroacetic acids, can have effects in biological assays. You also should think about the chemical features. If you're using a fluorescent assays, some compounds may have intrinsic fluorescence. Some may have reactive groups that interfere with your assays. Some may also be unstable, and some may have redox activities and oxidize components of your assay system. Another very common origin of false positives is compounds which induce unfolding or aggregation of biomolecules. And this is illustrated a little bit on the right side. If a compound induces aggregation, it will form these aggregates in your assay system, as shown in blue. And this may lead to the sequestration of your enzyme, of your protein of interest. The enzyme is not going to be able to function anymore, but this is not due to an inhibition, a specific inhibition that you want, but really only to the fact that it's trapped into these aggregates. Now, there are common warnings which can help you identify false positives. First, and we've discussed that before, compounds containing Payne's motif, pan-assay interference compounds, or reactive groups. Frequent hitters, so compounds which have hit in many previous screens, are usually interesting to flag because they're either false positives, compounds which usually interfere with assays, or they can just be promiscuous compounds, and these are usually also non-desired. Compounds with high lipophilicity have a tendency to be false positives. And singletons, for which no close analog is active, is usually also a sign that this can be a false positive. Not necessarily, but of course it should be a flag. And that's the reason why it's extremely important to use complementary assays to remove these false positives. And as mentioned, counterscreens, assays in which desired hit should not be active, or orthogonal assays, assays with an alternative readout in which the desired hits should remain active. Finally, and this is a concept that is extremely important in hit generation, to be able to demonstrate target engagement, binding of your compound to the target using, for example, biophysical methods is extremely powerful. So let's go into how we can filter compounds. As mentioned, we often do that based on compound structure, frequent Witters, non-interfering compounds with a specific technology like fluorescence, covalent binders containing reactive moieties. Paint filters are available online that you can use, and you have links to these on the bottom right side of this slide. And some companies like Eli Lilly have developed their own filters based on structural properties. You can also do this based on PhysChem or sample properties. We mentioned that purity, identity, solubility, aggregation, lipophilicity, or molecular weight. Now, one of the questions that very often comes during a screen, during the validation process, is do we filter out compounds, meaning we discard them, or do we just flag them for later? Now, obviously, if you filter them out, this will speed up the process, and there's anyway a low chance that these compounds are interesting. But in some cases, flagging only may be justified. For example, for difficult targets, if you only have a few hits, you risk to miss them. It's always target dependent. We talked about reactive compounds, but if you're actually looking for covalent compounds, then you for sure should not discard them. And the flags will not discard the compounds, but they can still be used to prioritize profiling in the cascade. So this is always a discussion and should be done case by case. Now, one thing that we should aspire to is to, in the end, generate the best screening library, where we filter out undesired compounds prior to screening so that we don't even have to care during the validation process. And this will save us time and resources. Okay, let's now go into the assays. And this slide explains a little bit the concept of the counterscreen, which is used to eliminate false positives. This can be done because you want to eliminate false positives due to assay interference and, as mentioned before, aggregate formation. And there you can, for example, use the same assay conditions and the same readout, but using another target. You can also use detergents to remove aggregators. What you also want to do is eliminate false positives or undesired modes of actions for a specific assay or target. For example, redox activity assays can be run to discard compounds that would inactivate an enzyme through oxidation. If you're running a cellular assay, it's almost a must to run a cytotoxicity readout. If your assay measures protein or gene downregulation in particular, cell death will actually lead to the same effect. And of course, you don't want to have a cytotoxic compound. You can also use, and this is illustrated on the upper right, an alternative stimulus for cell assays to remove unwanted mode of actions. So if your primary assay uses a stimulus that leads to pathway activation where your target of interest sits, you can use as a counterscreen a stimulus which will lead to the same pathway readout, but through a pathway where your target of interest is not present, meaning the compounds that you find here will not be interesting. Finally, it can be of advantage to assess early the selectivity on related proteins. Now, this is not a true filter because some compounds which are not selective may still be of interest, and you may build in selectivity later. It depends a little bit on the safety concern. For example, if you're working on a kinase, there are lots of kinase panels available, and you can run one to prioritize selective scaffolds and exclude compounds with safety risks. So on the right side, you have here a typical kinome scan tree where each end of the tree is a different kinase, and the size of these balls is the activity of the compound on a particular kinase. And when you have a profile like this one, basically hitting many kinases with very strong activity, that's probably not a compound you want to carry forward. Finally, it's worth mentioning that counterscreens can also be run in parallel to the primary assay or even built in the same assay. The prime example is cytotoxicity. You can build in a cytotoxicity readout within your primary assay. You can also use historical data to filter out hits. So, for example, you're working on a cysteine protease. If you've run a screen on another cysteine protease in recent years, you can use the hit list to filter out compounds which were active on both if this is not desired. The second type of assays is the orthogonal assays, where we want to confirm the activity of the compound in another system. Now, this is very important because when we confirm that the hit is active in another system, we really increase our confidence that it's working on the target of interest. Now, depending on the primary screen, there are different ways to do that. If you had a biochemical primary screen with a functional readout, maybe you can use a cell assay if you're working on an intracellular target, keeping in mind, of course, that the hits may not score due to low permeability. As mentioned, a very powerful method is to use biophysical methods to demonstrate target engagement, binding to the target. And you can do this with SPR, NMR, and we'll go into a lot more details in the next webinar. If neither of those is possible, you can use another biochemical assay with an alternative readout. For example, if you use the fluorescence assay, you can use a mass spectrometry-based assay. If your primary screen was a cellular assay with a functional readout, then why not run a biochemical assay if it's feasible? Here again, you can think about target engagement readouts, preferably in cells, and there are technologies called CETSA and others that have been developed recently to do exactly that. Or you can use an orthogonal cellular assay, and this is illustrated on the bottom right part of this slide. It can be, like in panel A, a different endpoint using the same cell line. So if you looked at gene expression, maybe this time you can use a functional readout. Or, like in panel B, you can use a different cell line looking at the same readout. If you used, as commonly done, a recombinant cell line in your primary screen, you can this time use a primary cell line. And if you have a consistent effect, that's a very strong sign that your compound is of interest. Now, it's important to note that the type and number of orthogonal assays selected to really build a chain of evidence for the hits is really dependent on the target, and it can be adapted based on the data. Sometimes one is sufficient, sometimes you may need more. Also important, some hits may not score in all of the assays and still be validated. So you really need to carefully analyze the data package and make decisions based on this. The last type of assay that we would like to mention are secondary assays. The idea here is to demonstrate as close as possible disease relevance. Again, to enable the high throughput, the primary and often the orthogonal assays are a highly simplified system where we try to focus on the target or pathway modulation. If we're able to confirm in disease-relevant functional assays, then we'll be in a better position to select compounds with a higher chance to later translate into therapeutic efficacy, first in animals and then in human. As an example, some mode of action will modulate the target as wanted, but this may not translate into what you actually want to do on the disease. So at the bottom is an example where we can measure muscle contractility in co-culture of rat motor neurons and human skeletal muscle cells. And here we have a little movie where you can see actually the muscle cells contracting. And in the graph on the left, you can see that by adding a GPCR agonist, we can increase contractility of the muscles by image analysis, and that this is reversed when we add an antagonist. This is actually nice because here we have a built-in additional readout which confirms the on-target activity. Now, the last topic we would like to briefly go through is ACR expansion based on your archive, your compound archive. The idea here is to test readily available close analogs. This is extremely important and almost done by default in any screening campaign. If you screen close analogs, you will increase the confidence in the hits if similar compounds are also active. You will get initial ACR and identify initial ACR trends like inactive compounds, ACR cliffs, ACR shape. You can also use that to do a counterscreen by ACR. If you have close analogs that are not active and you don't understand really the ACR, then probably the activity is not specific. It becomes critical if only a subset of the library was screened, because you have opportunities to find more attractive starting points. And if you remember the last webinar, this is more and more often the case. You can even source additional compounds from commercial sources to expand your ACR. All of that will increase in the end the confidence in the hit and make it more attractive for you to invest chemistry. We come now to the end of this webinar. Please don't hesitate to leave us comments or ask questions. We'll be happy to get back to you.